thanks for the invitation of the organizers. This Congress is really well organized. That's included the weather, everything is perfect. So this is my first time to visit your great country, and I saw in a couple of days, last couple of days, I was in Istanbul. It's a beautiful city and very kind and friendly people. So that's actually amazing. So I'm from Canada. So from Manitoba. Manitoba is in the middle of Canada. So you can see also in the middle of nowhere. So Canada is big. Now the background is our campus, our university campus. So we have a very beautiful river, it's called Red River. It flows through our campus. So I'm primarily with the Human Anatomy and Cell Science department, but I also have cross appointment with psychiatry. That's why I have some connection with the psychiatry people, particularly the organizer this time, Dr. Simi Lee, and we share a lab for several years. So we work together, and some of the work actually stems from us collaboration. See. So we know that in our brain we have billions of neurons, while all the neurons are organized in a kind of neuronal network. See, just Dr. Ball mentioned about the neuronal network. So in our hands, we can roughly divide this network into two parts. One, we call it hardware. The hardware actually, you see the cell body and the axon or dendrites, those form the hardware. And we also have the software part. That's all the neurons are connected by synapses. While the synapses, you have transmitters or modulators that Dr. Ball just mentioned. So she focuses more on the software part of the brain. And right now, I just want to mention that actually any part of the network, no matter it's the hardware or the software part, if it's not if it's damaged or not well organized, then you will have brain problems. So in the hardware part, in particular the axons, which actually is very complicated. Axons, neuron axons also wrapped with myelin sheaths. So today, I'm going to focus more on the myelin sheath, on how the myelin are formed. See, so the myelin sheaths in the brain are extremely important because it speeds up our neurotransmission, with also provide protection for the axons, and in some part of the brain, it's also very important to provide those nutrients, through the many of those deliver some of the nutrients to the axon, because the axon is very long and very far away from the cell body and need many of the metabolites supplied by the myelin sheath. So myelin is important. Myelin is also very dynamic, because it control or modulate the transmission speed. So in a way that there's a hypothesis mentioned that the learning process in the brain is actually the modification of your axon thickness or the myelin sheath thickness. So for example, if you have the signals that's transmitted from neuron A to B, before the learning process, because all the signals are not synchronized, whereas the learning process is to synchronize those neuronal action potentials so that they can reach to the secondary neuron at the same time, so that you have your new knowledge synthesized in the brain. See. So on the other hand, if the myelin is damaged or myelin is broken, then you have problem. Actually, this demyelination or dysmyelination, this happens almost involved in all our neurological disorders, including some of the psychiatric problems. Quite many years ago, there is a hypothesis about neurological disorder, including major depression, also schizophrenia, they have the so-called demyelination hypothesis. They say that the hardware part, or the axon, when the thickness of the myelin is changed or is not well organized, then in that situation, then you have transmission problem that's linked to many of the psychosis or symptoms. Say, for example, the multiple sclerosis, which is a predominant neurological disorder, but in the majority of patients, they do show some psychosis or some psychiatric problems. See, so in our lab, we focus more on the myelination, how the myelin is formed, in particular at the very beginning, how the myelination is initiated, what's the signals to trigger or to induce myelination in the last several years. So in general, what we know is actually myelination in the central nervous system is performed by a cell type we call oligodendrocytes. Well, oligodendrocytes initially comes from the neural stem cells. Normally, neural stem cell, you can find it in the subventricular zone or in some certain region of the brain, where those neural stem cells, they can be differentiated into the oligodendrocyte precursor cells, or OPCs. While these cells, once they differentiate into OPC, they can migrate along with the blood vessel, sometimes also with other structure, including the pioneer fibers of astrocytes, to a certain area of the brain to myelinate those axons. So that's the central nervous system myelination process, what we know about. Well, once the oligodendrocyte precursor cells move to axon, then the axon, on the surface of axon, there are many cues or many molecules. They can either be attractive or just to be repulsive. So those attractive cues, that's to attract those processes of the oligodendrocytes to make contact with the axon and to wrap with the axon, then to spread on the axon to form the myelination process. Well, of course, on the other hand, we have many inhibitory molecules which can prevent that from happening. See, now that's a model actually has been suggested for many, many years. Well, in general, it's true, but many of the mechanisms that's involved in the myelination process, still we know very little about those processes. We know that sometimes these factors, including the transcriptional factors or signaling pathways or axon signals, including some of the those extracellular factors which participate in this process. But if you really want to know or to ask what's the factors that trigger this process, this myelination process, we really don't know. But we do know, actually. In the brain, we have 4 types of cells. Neuron predominantly, and we also have glial cells, including astrocytes and oligodendrocytes. Those are the neural cells. In addition to those neural cells, we have another type of cell that's microglia. So there are studies suggesting that microglia are involved in the myelination process. One word, without microglia, there's no myelination. So this has been demonstrated. If you knock out or just delete the microglia, then in the brain of this animal, they don't form myelin, or the myelination is significantly affected. Now, in our study lab, we developed a system that's in vitro myelination. Actually, it's very simple. So if you just do the brain cells or spinal cord cells that include the neurons and astrocytes and microglia, all the cells mixture, you just do a cell culture after several days, like 20 days, then you start to see myelination. 25 days, you see lots of the myelin going on. So that's a mixture of the brain tissues to its culture. However, if you differentiate those cells from your stem cell, then so we know neural stem cell can be differentiated into neurons, astrocytes, and oligodendrocytes. But those cells, technically, they have new axons, they have oligodendrocytes. They should be able to form myelin. However, on the right side, you see, no matter how long you put it in the tissue culture, those cells don't talk to each other. So the oligodendrocytes and axon, they don't talk. But if you put microglia into the tissue culture, immediately you can induce those like a process of oligodendrocytes. They can make contact with axons, so form those cell-cell interaction right away. So from this experiment, we can conclude that without microglia, you don't see myelination. So microglia has to be a factor that trigger the myelination. So if you do a careful analysis, so in the mixed culture, that's brain tissue-derived cell culture, you have about 10% of cells actually as microglia. Or if you do the neural stem cell culture, there's no microglia, because microglia come from different type of stem cell. While neural stem cell can only differentiate into oligodendrocytes, astrocytes, and some of of course, neuron and some of the other cells. See, now from there we know that microglia is required, but then we did another experiment. We don't put microglia into the tissue culture. We put the microglia conditioned medium into the tissue culture that can still induce myelination. What does it mean? That means that actually just not the cell body is required, but just the secretion of microglia is required. We know microglia can secrete a lot of chemokines, cytokines. On the right side, you see lots of them. Some of them are they can promote proliferation. Some of them actually inhibit proliferation. See, we know at this point we conclude that actually the secretome or the secretion of microglia, which is important for this one. But which one, actually which molecule, is really play a role? That's a big story. Now we did a lot of the selection or lots of the antibody neutralization study, and spent several close to 2 years, and figure out one molecule, the molecule called C1q, which actually is important in this process. So C1q is the old molecule, has been studied for many, many years. Its primary function is in the immunological study has found that actually it's important in immunity. We found that actually C1q is a factor that triggers the myelination, because see, if we do a Western blot, we found that in brain tissue, you can find the C1q there. Or if you use the neural stem cell culture, you don't see any C1q. If you do the cell culture from the spinal cord, mixed culture, then with time going on, you start to see C1q expression. So at the very beginning, you don't see C1q, but 2 weeks or 4 weeks or 6 weeks later, you see lots of very high level of C1q expressed. Now, here you see, if we put C1q on the right side, that's a mixed culture, spinal cord tissue culture, you can see very beautiful myelination. But if you remove the C1q from the stem cell culture, then there's no myelination. The second one is the stem cell culture. Obviously, there is no microglia, so there's no C1q. On the left side, if you neutralize those or remove C1q from the mixed culture, then you can just abolish the myelination process. Okay. Now, we did another experiment. We just put the C1q from Sigma, just commercially available C1q, put it into the neural stem cell culture. Then we can induce myelination in a concentration-dependent manner. See, about about 100 micromole C1Q, that's human C1Q put into animal, into mouse neural stem cell, you can trigger myelination. This suggests that C1Q is not just necessary, but sufficient to trigger myelination. Now, we did that's an in vitro study. We want to move on to in vivo validation, and we found out that C1Q is expressed at very early level. We know in animal, the myelination starts by P7, so 7 days after birth, you see myelination in mouse model, while C1Q is expressed at birth already. Now, the expression pattern and the myelination pattern correlate very well. Okay, what I mean very well, see, we know myelination in the brain happens at the brainstem part, then go rostrally and posteriorly. Now, C1Q expression pattern or the profile is correlate very well with the myelination process. Now, that's just correlative study. Then we further went on and we did the conditional knockout animal model to knock out C1Q. Because complete knockout C1Q is lethal, we can't do anything. Once you knock out C1Q, the animal simply die. So we had to do those conditional knockout. We allow the animal to express C1Q. Then after birth, from day 3, we introduced tamoxifen in the system. Then the animal, see, basically, we just to reduce the expression of C1Q, we did not wipe out C1Q. C1Q still expressed in low level, but reduced significantly. In that case, then we can induce actually this hypomyelination. So myelination is reduced significantly. If you see this picture, on the right side is normal. On the left side, actually that's a C1Q conditional knockout by P10. You see a significant reduction in the myelination process. So from this, we know that this experiment is not perfect because still you have a certain level of C1Q expression, but we can already see a significant reduction of myelination. See, from there, we found that actually C1Q is a major factor in the myelination, in triggering myelination process. Then we want to know what process, actually which step is C1Q involved. So we did a different time, like injection of the tamoxifen into the animal to induce conditional knockout process. And what we found actually, C1Q knockouts does not prevent or does not change the expression level of like a mining. In other words, knockout C1Q does not affect the production of mining. But what it does is actually it changes the alignment of mining or assembly of mining into those longitudinal or those sheath, mining sheath. So it's without C1Q, the mining cannot be organized into those mining sheath. That's a conclusion. So from there, from those evidence, we conclude that C1Q is required and sufficient for CNS myelination. And C1Q expression profile, which is correlated with myelination patterns, and conditional knockout C1Q leads to hypomyelination. So that's our conclusion with that. So we say that actually C1Q does not affect the production of myelination, but affect the assembly of mining into mining sheath. Okay, that's our conclusion. See, I have to acknowledge that my lab has been in collaboration with many labs worldwide, including someone sitting in this room, like Dr. Xu from Suzhou University and Yu Tianwang from UBC, and similarly my longtime collaborator. Okay. And my research is supported by lots of national and international grant agencies, and particularly recently we received some private donations. They just give us money without any grant writing, which I don't like. So that's the people who did this work from this mass stroke project. Actually, that's their name in red. Did this work, and I also have a team. We are working on other projects that's including the aging and the neurodegeneration project. With that, thank you for your attention.